Hi everyone, this is the first mini lecture of chapter 13. We'll start with the learning outcomes. I'll go ahead and let you guys read these and you can pause it here if you need to take some time, but I'm going to move straight into the lecture. So we're talking about specific immunity, the third and final line of defense. And this is acquired immunity because it happens after an immunizing event, meaning that it occurs after an infection or some sort of exposure to an antigen. And this can take place because the body has the ability to react to foreign substances. We call this immunocompetence. The two really special features that characterize the specific immunity is specificity, meaning that there's recognition of a particular antigen or immunogen, and that there's memory, meaning that if we are exposed to the same antigen a second time, the attack will be greater and quicker. With specific immunity, we need to have antigens or immunogens that can stimulate a response by B and T cells. So these would be something about the infectious agent that would stimulate an immune response. The portion of that antigen that actually describes it as foreign is what we would call the epitope. That's just a portion of the antigen. And generally speaking, it can be protein or polysaccharide molecules that are on or inside cells, prokaryotic cells, and viruses. And it's any exposure or release of the protein that can possibly be an antigen. And as you remember probably from the second line of defense, we had PAMPs that were to flag these kind of foreign invaders. In this case, antigens are actually individual and will stimulate the specific immunity, not that generalized second line of defense. What we generally see, or the principal stages of that immunological development, so how these cells come about, is we have lymphocytes that develop and differentiate from that original stem cell, and it's through the presentation of antigens that can challenge the B and T cells. With the T cell lymphocytes, what we see is a cell-mediated response, meaning that when a T cell is stimulated, it will stimulate other T cells to attack this invader or to increase the immune response to the invader. With B cell lymphocytes, we get production and activities of antibodies, so they're a little bit different. B cells are actually in charge of getting antibodies to fight the particular antigen. So, as I said, the lymphocytes will come from that basic stem cell type. B cells, we want to always keep in mind that they mature in bone marrow sites, and T cells mature in the thymus. And these B and T cells are found in the circulatory system and lymphatics, but they're constantly circulating, and that's on the lookout for antigens that might be coming into the body. This is showing from that stem cell that we would start with, and then how they develop with and without the antigen response. So at the top, we have that lymphocyte. T cells will go to the thymus and will mature here, and then they'll hang out in the lymph nodes or the spleen, and they'll also be circulating in the circulatory system. With the B cells, they mature in the bone marrow, and then once again we see them in the lymph system and in the circulatory system. And where we get that stimulation is when we have antigen. And so in this case, it's showing this little blue antigen up here. This could be a virus or a bacteria. And when it comes in, it's going to be phagocytized. Remember, from that second line of defense, it'll be broken up in some sort of phagocytic cell. In this case, they're showing a dendritic cell. When it's broken up, this cell has the ability to take a portion of it, the portion that can stimulate an immune response, and present it on its MHC, major histocompatibility complex, on the outside of that immune cell. And this is special because when it's presented in this way on a self cell, a T cell will recognize that antigen. It will actually bind to the antigen, but also to the phagocytic cell. And this is the way that it recognizes that this is an invader that it needs to respond to. And so in this case, the T cell here then is activated, and this T cell can then go on to form memory T cells to promote the addition of memory T cells. It can also go on to become a specialized effector cell, whether that's a T helper cell, a T regulatory cell, or a T cytotoxic cell, which are the ones that can actually kill. So this is what happens on the T cell line. We'll get into it a little bit more detail later in this chapter. But this T cell also, once it has been stimulated, can stimulate the B cell line. And these B cells then, when they're activated, will also form memory cells, but then they will form plasma cells which can secrete antibodies. And when we get further into this chapter, you'll see the role that antibodies play, but they are going to be specific to the antigen that originated up here. So hopefully that makes some sense, and we'll get a little bit more into detail as we move forward in this chapter. So I spoke about the major histocompatibility complex. This is a set of genes that codes for these cell markers or receptors, and they are found on all cells except for red blood cells. And they play a vital role in recognizing self by the immune system. So the immune system will recognize that that cell is a self cell, and also will check to see if it's a different type of molecule that it would be foreign tissue because it doesn't have that major histocompatibility complex. Within the major histocompatibility complex, we have 3 different classes of genes, which means 3 different classes of this MHC molecule. The class 1 genes are going to code for markers that will appear on all nucleated cells. And in this case, they are for recognition within the immune reaction. If you are showing this class 1 MHC, immune cells say, okay, I know you, you're a good guy, we're not going to go after you. In the case of class 2 genes, these code for immune regulatory markers that are found on macrophages, dendritic cells, other cells of the immune system, and they're the ones that are capable of presenting these antigens to the T cells. So in the case of class 2 genes, when that microbe, whether it's a virus or a bacteria, has been degraded, it's actually the class 2 MHC complex that will present that antigen to the T cells. And then the last one we have is class 3 genes, and these have the proteins that are involved with the complement system, which you hopefully remember from chapter 12. So when we have an antigen that has been presented by an MHC2 complex, both the B and T cells are going to start to proliferate or to make duplicates. And they will also start to differentiate. Remember, we talked about some of them can be memory cells and some of them can go on to be other things. So in some cases, they could go on to be a clone or a group of genetically identical cells. Some of these clones will be memory cells, and that's so that we have that memory if we get exposed to this antigen a second time. And keep in mind that the B and T cells responses differ. Remember, the B cells are going to be producing antibodies, and the T cells are cell-mediated, meaning that they are going to proliferate into T cells that can aid in this immune response. So when we have that antigen exposed and a T cell recognizes it, what the T cells will do is they will begin to proliferate. They can proliferate into helper T cells, which will aid in the immune response, whether it's activating macrophages or helping with the B cell processes, or even help activate cytotoxic T cells. They can also proliferate into regulatory T cells, which can control that T cell response, either tamp it down or ramp it up if needed. And then last, they can proliferate into cytotoxic T cells. And these are T cells that can destroy the infected host cells and other foreign cells. So these are one of our ones that can actually destroy the infection and the infected cells. T cells also have the ability to secrete cytokines. Remember, these are chemical signals that can play a role in immunity, but they do not produce antibodies. That is only B cells. So how do those B cells respond to the antigen? When B cells are activated, they are going to divide. Some of them will become memory cells, And then some of them will give rise to plasma cells. And it's these plasma cells that have the ability to produce antibodies and release these antibodies into the tissue and blood. Antibodies are really special because they can attach to the antigen that they are specific for, and then that antigen will be marked for either destruction or neutralization. And as we get into some of the other mini lectures in this chapter, we'll talk about how those work. So the last little bit on this is to check in. These are all things that you should be able to answer. If not, you can go back and look at this mini lecture. If you have any questions on this, please let me know, and I will be continuing with the next 5. Hopefully this is helpful. Thanks.